Biotechnology. There is a great scope in this branch of science, and let us examine it. Biotechnology, the definition given by European Federation of Biotechnology, that is EFB, is the integration of natural science and organisms, cell parts thereof, and molecular analogues for products and services. Now look here. These are the various horizons where biotechnology is of great use nowadays. Say, for agriculture, people are developing improved varieties, pest resistant. Then, of course, medicine. You must be knowing that now for the diabetic patients, the insulin is being synthesized by the bacteria and which is consumed by the diabetic patient. Then, of course, you have chemical industry. You are just producing chemicals which are of great use to our daily life by biotechnology. Food industry, we have now developed certain genetically modified plants which are called as GMO, like your BT cotton, BT maize, BT soybean. Then, of course, environmental protection. You must have heard that there is a great oil pollution. Dr. Chakravarti has developed the genetically engineered bacteria, that is Pseudomonas putida, which has the ability to consume all the types of hydrocarbons, and that is how, if you introduce this bacteria where oil spill has taken place in the ocean, the oil pollution from the ocean will be removed. And then, of course, you have the power industry and many more. Two core techniques are involved in the modern biotechnology. Number one is genetic engineering, altering the chemistry of genetic material, that is, of course, DNA and RNA, to introduce it into the host organism and thus change the phenotype of the host organism. Now, here you can see that I just told you while discussing the biotechnology scope, where the BT gene has been introduced in the corn plant, and thus the corn plant has developed resistance to corn borer. So this is a BT gene introduced in the GMO, and the BT corn is a genetically modified corn. Maintenance of sterile, contamination-free ambience in chemical engineering processes to enable the growth of desired microbe eukaryotic cells at a large scale to produce biotechnological products like enzymes, proteins, antibiotics, etc. As you are aware that the sexual reproduction permits variation, and this variation is due to crossing over which is occurring during the meiosis. But the only drawback is that sometimes undesirable genes along with desirable genes also gets incorporated. The technique of genetic engineering overcomes this limitation and introduces only the desirable gene in the target organism. Stanley Cohen and Herbert Boyer, 1972, were the first to construct the recombinant DNA by linking a gene encoding for resistance of antibiotics to the plasmid of Salmonella typhimurium. So they can be referred as the pioneer in the field of DNA recombinant technology. That is how they introduced a gene encoding resistance to antibiotics in Salmonella. The separation of antibiotic gene from the plasmid was possible after the discovery of the enzyme restriction endonuclease. Arbour postulated the existence of this enzyme in 1962, while Smith and Nathans isolated the enzyme from the bacterium Haemophilus influenzae and named it as HIN2 This restriction enzyme is also called as biological scissors or biological scalpel. Three steps are involved in the genetically modifying organism. Here you have the photograph of the Haemophilus influenzae bacteria. First, the identification of the DNA with desirable gene. Introduction of the identified DNA into the host. Maintenance of introduced gene in the host and transfer of the same to its progeny. The key tools involved in the genetic engineering are restriction enzyme, polymerase enzyme, ligase, vectors, and the host. Restriction enzyme, that is HIN2, always cut DNA at a specific point by recognizing a specific sequence of 6 base pair, that is CTTAAG, called as palindromic nucleotide sequence, as recognition sequence. Each restriction enzyme recognizes a different recognition sequence. Now here you can see that is this palindromic 6 bases is there from where this HIN2 cuts the DNA. Restriction enzymes are referred as nucleases, and they are of two kinds. Number one, exonuclease removes the nucleotide from the ends of the DNA, and here you can see that the single strand nick is there, and it is from one end the DNA is cut. That is the exonuclease activity. Number two, endonucleases cuts the DNA at a specific position within the DNA. And here it is within the DNA the segment is cut. The restriction enzyme cuts the DNA molecule by cleavage in any of the following ways. Number one is the blunt end. Restriction enzymes like HIN2, ALU1 make cuts across both these strands of DNA at the same position sequence recognized. This is referred as blunt end. You can see here the DNA segment has been separated and the end is blunt. Number two, staggered cutting or sticky end style. Here the cuts are made in two strands of DNA at two different sites, few nucleotides away from each other, thus producing two strands with sticky ends. Here this is the staggered or the sticky end. And this is the palindromic sequence. You can watch it, GAATTC, and on the other it is GAATTC. So it cuts between A and G. The restriction fragment of DNA isolated from one organism will have the same sticky ends as the restriction sequence from the other organism, provided they are produced by the same restriction enzyme. Separation and isolation of the DNA fragments. The DNA fragments obtained by the activity of restriction endonuclease can be separated by gel electrophoresis. The DNA is negatively charged, so they will move towards the anode under the electrical field through a medium or matrix. And you know that the most commonly used matrix is the agarose obtained from the seaweeds. That is the red algae. The DNA fragments separate by the sieving effect of the agarose gel. The separated DNA fragments can be visualized after staining with ethidium bromide and then exposing to UV radiation. It appeared as bright orange colored bands of DNA, and this process is referred as elution, and this you can see here in this diagram. This is the gel electrophoresis. You can see here, and then in the second diagram, staining of the DNA with DNA binding dye, that is ethidium bromide. So by ethidium bromide, you can visualize the DNA when exposed to UV light. Cloning vectors. Plasmids and bacteriophages have the ability to replicate within the bacterial cell which is independent of the control of bacterial genome. The alien piece of DNA is linked with the DNA of the bacteriophage or plasmid, and then we can increase the number of alien DNA equal to the number of plasmid or bacteriophages. The vectors used now are modified in such a way that it facilitates easy linking of foreign DNA and making it suitable in the selection of recombinants from non-recombinants. Now here you can see that is how the foreign DNA from which the DNA insert would be extracted. Then you can see that target DNA segment has two restriction sites, hence there are two nicks. And then you can see that is how it finally, a chimera is now formed. That is, that foreign DNA has been incorporated in the plasmid vector. The following features are necessary to facilitate the cloning into a vector. Number one, origin of replication, that is ORI. To attach the DNA fragment to that place from where the replication starts, and thus it can be made to replicate within the host cell. Here you can see the origin of replication is there. That is single-stranded DNA binding proteins. Helicase is there, primase is there, and this is the point which is referred as ORI. Next is selectable marker. Besides ORI, the vector needs a selectable marker which helps in identifying and growth of transformants and eliminating the non-transformants. Transformation is a procedure where the piece of DNA is introduced in the host bacterium. Some of the useful selectable markers of E. coli are gene encoding antibiotic resistance genes such as ampicillin, chloramphenicol, tetracycline, or kanamycin, etc. Now here you can see that is these are the selectable markers. This is a vector which is PUC18. Then comes the cloning site. In order to link the alien DNA, the vector should have few but preferably single recognition site for the commonly used restriction enzymes. The ligation of alien DNA is carried out as the restriction site in one of the two antibiotic resistant genes. For example, you can ligate a foreign DNA at the BAM-H1 site of tetracycline resistant gene in the vector PBR322. You know that PBR322 is a created vector. You can see here, this is the PBR322 vector, which is commonly used in the genetic engineering. A standard cloning vector obtained from E. coli, it is a genetically engineered and has one or two genes for resistance against antibiotics. The recombinant plasmid will lose tetracycline resistance due to the insertion of foreign DNA, but can be still selected out from non-recombinant ones by plating the transformant on ampicillin containing medium. The transformants growing on ampicillin containing medium are then transferred to a medium containing tetracycline. The recombinants will grow on ampicillin containing medium but not on tetracycline. On the other hand, non-recombinants will grow on medium containing both the antibiotics. Selection of recombinants due to inactivation of antibiotics is a cumbersome process. It takes a lot of time because it requires plating simultaneously on two plates. Now they have developed another technique or alternative selectable markers to distinguish recombinants from non-recombinants on the basis to produce color in presence of chromogenic substance. In this technique, recombinant DNA is inserted within the coding sequence of an enzyme, for example, alpha galactosidase, and this results in the inactivation of enzyme, and it is referred as insertional inactivation. The blue color of the colony by chromogenic substance indicates that it does not have the insert, and colonies do not take color, and it will be identified as recombinant colonies, and this can be seen with the help of this diagram. You can see here that is how the insertional inactivation is done. Here you can see you have the foreign DNA insert, then you have recombinant plasmid, ampicillin resistance is there, transformation, foreign DNA enters the host cell, selecting cells that have received a plasmid. And then you have distinguishing cells carrying insert containing recombinant molecules from cells carrying vectors without inserts. And that is how it is done. Agrobacterium tumefaciens. You know, this is a bacteria which causes disease in dicot plants. The tDNA of the bacterium transforms the normal plant cells into tumor cells. Similarly, certain retrovirus transform normal animal cells into cancerous cells. It is now possible to transform these pathogens as useful vectors for transferring genes of interest in eukaryotic cells. And here you can see the tDNA and the Ti plasmid. This is the bacterial chromosome in the bacterial cell. Agrobacterium tumefaciens have been modified into a cloning vector, which is no more pathogenic, by removing the tumor-inducing gene from Ti, that is the I gene, from the plasmid. Similarly, retroviruses are transformed and now in use to transfer desirable genes into the animal cells. Competent host, that is for transformation with recombinant DNA. DNA is a hydrophilic molecule and cannot enter through the cell membrane. The conditions are created to make the host cell, that is the bacterial cell or maybe eukaryotic cell, competent to take up the DNA. And here you can see that the foreign DNA is being entered with the help of the receptor. A number of techniques are used to allow the entry of the alien DNA into the host cell. Treating the cell with a specific concentration of divalent cation such as calcium, it stimulates the entry of DNA through the pores in the cell wall. This is number one. Number two, incubating the cell along with the recombinant DNA and ice, followed by placing them briefly at 42 degrees centigrade and then back to ice. Recombinant DNA is directly injected into the nucleus of host cell, and this is applicable in animal cells. Use of biolistics or gene gun. You can see the operation of the gene gun here, and this is suitable for the plant cell. Here the microparticles of gold and tungsten coated with DNA are bombarded into the plant cell, and you can see that how gene gun is operating and inserting these microparticles into the target tissue. And below that is the gene gun with the plant. Processes involved in DNA recombinant technology. Let us revise. Isolation of desired DNA segment, that is gene, by restriction endonuclease. In order to achieve, we treat the bacterial cell, maybe animal cell, with enzymes like lysozyme, used for bacterial cell, cellulase for plant cell, chitinase for fungi. The purified DNA ultimately precipitates after the addition of chilled ethanol. Agarose gel electrophoresis is done to separate the DNA fragments. Amplification of the gene of interest using polymerase chain reaction, which is short called as PCR. Multiple copies of the gene, that is DNA, is synthesized in vitro using two sets of primer, that is short segment of RNA synthesized by RNA polymerase, DNA polymerase, nucleotides, and the genomic DNA as template. The enzyme DNA polymerase used in the PCR technique is isolated from the bacteria Thermus aquaticus. This bacteria's photograph is before you, and this DNA polymerase is heat resistant, and the amplified fragments can be ligated to vector for further cloning. And this is how the PCR, that is polymerase chain reaction, is done with the help of enzyme DNA polymerase isolated from the bacteria Thermus aquaticus. Obtaining the foreign gene product. In DNA recombinant technology, the ultimate goal is to produce a desirable gene on a large scale. They may be grown on a small scale in laboratory. The cultures may be used for extracting the desired protein and purify it using different techniques. In order to produce a desired gene on a large scale, the bioreactors are used. And the most commonly used bioreactors are string type. And this is the diagram of the string type where you can produce the desired gene or the product at a large scale. Downstream processing. As soon as a biosynthetic stage is completed, the product is further subjected to a series of processes which include separation, purification, and these are referred as downstreaming processing. And this is done in this way. You can see you have the upstream processing, then you have the fermentation, and then you have the downstream processing, and ultimately you are getting the product.